Okay, so welcome everyone. Today, regulatory genomics, motif discovery, target prediction, regulatory region dissection. We're entering a new module. We're going to talk about motifs today, and then how to understand the networks, and then some deep learning techniques in a couple of lectures. So first, we're going to talk about what are regulatory motifs. Then we're going to talk about two different ways of discovering them in co-regulated gene sets, expectation maximization and Gibbs sampling. Then we're going to talk about evolutionary signatures for de novo motif discovery. And then we're going to talk about evolutionary signatures for individual instance identification. And then we're going to talk about de novo dissection of regulatory regions using high throughput experiments. So let's dive right in. What are regulatory motifs? So regulatory motifs are the building blocks of gene regulation. So if you look at a region of gene regulation that I showed you in one of the first lectures, this is the GAL1 gene that metabolizes galactose in yeast. This is the GAL4 promoter that binds these CGG, spaced by 11 nucleotides, from CCG motif, which is a palindrome. And then this is the MIG1 regulator. So when these guys bind, they recruit a lot of machinery, which eventually converges on the promoter of GAL1, where there's a Tata box, and then that's where polymerase binds and initiates transcription and ultimately translation at the ATG. So genes are turned on and off in response to changing environments, and there's no direct addressing. So I don't know if you guys remember go-to routines in C and assembly and so on and so forth. These don't exist in genomes. The subroutines, i.e. the genes, contain within them sequence tags, which are motifs, and then there's specialized proteins, which are transcription factors that recognize these tags. And what makes motif discovery hard, as opposed to, say, gene finding, where you always have an ATG, you always have the same splice acceptor and donor, a branch site, and so on and so forth, and a particular codon pattern. What makes motif discovery hard is that motifs are very short. They're sometimes degenerate. For example, the MIG1 motif is CCCCW, which is A or T, a weak base with only 2 hydrogen bonds. They can contain any set of nucleotides. There's no ATG, there's no other rules. And they also act at variable distances upstream and downstream of their target genes. So that makes discovery of these motifs very, very hard. And motifs are everywhere. Motifs are, in promoter regions. Motifs are in enhancer regions, these distal regulatory regions that loop around to the promoters. Motifs are involved in splicing, both for the donor and acceptor, but also the branch side, but also within the exon and within the intron, there's exon inhibition signals, and there's exon, enrichment signals that basically cause exons to be included more or less frequently. So ESCs and ESIs and ISIs and ISCs are basically classes of motifs, which are, very degenerate and very similar to those motifs that act actually from within the protein coding sequence or from within the intron. And then there's 3' UTR motifs, which are recognized by microRNA. So what all of these have in common is that they are patterns, which can be at varying positions, and they can be degenerate. And that's what we're going to be focusing on today. So how do we discover motifs in co-regulated promoters, and also how do we discover motifs de novo and identify their targets? So transcription factors use DNA binding domains to recognize specific DNA sequences in the genome. So the way that this recognition happens is the factor actually binds the closed DNA double helix. It doesn't open up the double helix. It binds it closed, and it makes contact with specific residues, and the specific contacts that it makes define whether that motif will be, a particular shape or not, okay? So here, if it says T or C, it basically means that both T and C have the same atoms that the transcription factor likes to recognize at that position, and that's what causes the degeneracy. And same thing here, A or T, G or C, A or C or G, and so on and so forth. So we're going to be looking at a lot of these motif logos as a way to understand gene regulation and the foundation of gene regulation. And the sequence of the motif, in fact, is directly dictated by the structure of the factor. So, for example, the Tata factor here is bound contiguously. The CAG GTG motif is sometimes actually found spaced by some nucleotides. And the reason for that is that it's bound by a dimer. And this other motif is, again, bound by this transfer factor. And these regulatory motifs are hugely important in gene regulation. As we're going to talk about in a few days when we start the module on disease genetics, the vast majority of genome-wide association study hits that have come up are in fact sitting in non-coding regions. So 93% of genetic variants associated with complex traits are non-coding. And what they do is that they disrupt these individual regulatory motifs. To give you an example from our own lab, we basically studied the genetic signal underlying obesity in this FTO locus. And we were able to pinpoint the signal from this, very large, 50 kb region spanning 89 common variants in the first intron of this gene into a single nucleotide disrupted by this RS1421085 genetic variant, which changes a deeply conserved T into a C, leading these transcription factors to no longer bind. And that is actually at the core of new circuitry that we uncovered in that paper that basically shows that there's this AT-rich interacting domain factor that normally binds that motif to repress these genes. When that T is changed into a C for the obesity risk individuals, the repressor can no longer bind. The super enhancer becomes overactivated, and IRX3 and RX5, two downstream target genes, which are 1.2 million nucleotides away and 600,000 nucleotides away, are in fact de-repressed, and they turn off thermogenesis and they turn on lipogenesis, leading to a shift from energy burning to energy storing, from calorie burning to calorie storing, and ultimately from lean to obese individuals. So these motifs are at the core of disease circuitry in 93% of genetic association study results from 120,000 loci that are known to be associated with disease. 93% of them, more than 110,000 loci, remain to be dissected. This is one example, but there are many, many, many more examples that remain to be dissected, and regulatory motifs are at the core of that. So, as I mentioned earlier, RNA-DNA recognition happens by complementarity. You have to open up DNA, and then you feel the individual nucleotides, and then that leads to the sort of complementarity between DNA homologous strands, or between RNA and DNA through strand inversion of the double helix, and so on and so forth. But proteins never actually open up the DNA. They just read the chemical properties of the bases. There's no base complementarity, and instead, it's the 3D topology that dictates the specificity. There's fully constrained positions where every atom matters, and there's ambiguous or degenerate positions which are loosely contacted. And there's many other types of recognition, as I mentioned, microRNAs. Nucleosomes are primarily driven by GC content. RNA structures combine both structure and sequence together for recognition, and so on and so forth. So what do motifs do? Motifs summarize transcription factor binding specificities. So if you look at this particular motif, it's very intuitive. It basically says, oh, this position better be a T, that position better be a C, that position better be an A, and so on and so forth. And that position, well, it doesn't matter. It can be either an A or a G. But where do these come from? They come from summarizing information which is scattered across many locations in the genome. Here I'm showing you the ABF1 regulator in yeast, and what are the experimental footprints that folks have made for that factor. So what are these experimental footprints? You basically sort of have a region where this factor is bound. You protect the nucleotides that are bound by the transcription factor based on the binding of that factor. And then you digest the DNA from the outside to basically only leave that footprint behind. And that footprint gives you the specific positions of binding. And what's really cool, what really started this whole field, is that if you look at these regions, you basically see that, wow, this position appears to be either an A or a T or a G. And that position is always a T, that position is always a C, that position is always an A. So we can summarize this using a position weight matrix, or a PWM, that basically tells you for every nucleotide how frequently is that nucleotide found at every position. And you'll notice here in these intermediate middle positions, all nucleotides are found at equal frequency. So, it really doesn't seem to carry that much information, except for, well, this position better not be a G. Otherwise, all three positions seem to be, tolerated with T at a slightly higher frequency. So these motif logos basically summarize the position-specific matrix that tells you the frequency with which each of the nucleotides is found in experimental sites. And you can think of this as a generative model, where you can basically generate instances of a motif, and you're inferring the generative process through which this motif occurs through the logo. So the logo is a generative model, and these are individual instances from that model. And the height of these positions in the visualization of these motifs is a measure of information. It basically tells you how many bits of information do I have at that position. And here I basically have 2 bits of information, assuming a uniform nucleotide background. Why? Because it takes me 2 yes or no questions to obtain the identity of that nucleotide. Is it a purine or a pyrimidine? And if I know the answer to that, then I say, is it a G or a T, and so on and so forth. So that, that differs based on the background distribution, of course. If my background distribution is very AT rich, then seeing a T at that position contains less than 2 bits of information. If that background is very GC rich, seeing a T gives me more than 2 bits of information because the surprise level is higher, okay? So, all of this is directly driven by information theory, and that, that dictates the height of those motifs. So we're making several assumptions. We're making the assumption of independence, that every position can be independently defined from these other positions. For example, here, if I choose an A versus the G, that has no bearing on whether I choose a C or a T at that other position. So that's the position independence. Alternatively, it may have been the case that if I have an A here, I always have a C here, and then that's not position independent anymore. And the way to represent that is to split my motif into 2 motifs, one for the A-C combination with all of the other bases whenever A and C are defined together, and another one for a G and T whenever all the other bases are basically with all the other bases according to that. But of course, even that will not be, actually that will be a pretty close approximation. So most of the time we're going to assume position independence, and if you have position dependencies, then you can sort of split them up into different motifs. We're also assuming fixed spacing, and that's not always the case. So there are some motifs for which there's actually variable spacing in between the 2 parts, and these we simply won't be able to capture. But again, hugely important to distinguish the motif, which is the generative model, versus the instances, which are the individual occurrences of that motif. And what we're going to be using is those instances as a way to infer the motif as a latent variable, as a nuisance parameter that we're going to be iterating over in sort of trying to find all of the occurrences of that motif. Okay, so let's see who's with me so far. So we've talked about motifs, we've talked about sort of where they're coming from, how to define them, how to recognize them, and how to represent them using this information theoretic approach. Awesome. So we have 55, 45, 00. That's great. All right, so again, motifs are not limited to only DNA sequence. So splicing signals at the RNA level with splice junction, exonic splicing enhancers, exonic splicing suppressors, intronic enhancers, intronic suppressors. Specific domains and epitopes at the protein level, for example, glycosylation sites can be represented as motifs, kinase targets, targeting signals, the major histocompatibility complex binding specificities, and also recurring patterns at the physiological level can be represented as motifs. For example, gene expression patterns during the cell cycle. You could, sort of infer the patterns of cell cycling in the same way that we're defining motifs today. In fact, a final project in a previous year, which is actually now used in Boston hospitals, was to look for heartbeat patterns that are predictive of cardiac arrest. And one of the students in the class actually defined a project based on that. And, you know, frankly, any probabilistic recurring patterns can be recognized as a motif. So there's many approaches for discovering regulatory motifs. There's what I like to call region-based motif discovery, genome-wide motif discovery, and also in vitro or transacting, which basically means, you know, focused on the protein discovery. So region-based, we're going to look at two different approaches today. One is expectation maximization, which iteratively refines the positions of the motif profile. The second one is Gibbs sampling, which is, so we've seen expectation maximization before. We haven't seen Gibbs sampling yet. So this is a super, super important technique, pervasive in computer science, so definitely worth knowing. And this iteratively samples positions versus on the motif profile. There's enumeration, possibly with wildcards, and we're going to see one such enumeration approach in the context of genome-wide motif discovery. And then there's also correlation approaches that focus on whether the peak height of binding of transcription factor based on the ChIP-seq experiments like we talked about 2 lectures ago is in fact, you know, predictive of the motif agreement, basically whether the strength of the PWM matches the strength of the chromatin precipitation. So these are just different families of approaches, and we're going to focus on 3 of them today for region-based motif discovery. What's region-based motif discovery? That's if I have a set of regions, can I use them to define a motif that's common within these regions? The second type of approach is genome-wide motif discovery. That's basically saying we're going to use evolutionary conservation as a means of predicting that signal. And we're going to define genome-wide scores as well as, you know, upstream versus downstream biases and so on and so forth. And then the third approach is using experiments such as protein binding microarrays or these selection techniques for finding these motifs in vitro. So let's look at some of them. So basically, protein binding microarrays are basically building a set of k-mers so that every possible k-mer is represented and washing through proteins that are possibly recognizing those k-mers. And then what you end up with is the intensity telling you where each of these k-mers, where each of these proteins were bound. And then from these binding locations, you can say, okay, well, they predominantly bind, you know, this 8-mer within my 10-mers, for example, and then infer the motif that way. A second one is this systematic evolution of ligands by exponential enrichment, which basically is building an RNA library, washing it against a protein target of interest, and then partitioning the bound versus unbound RNAs, and then repeating the process. So you are basically at every iteration selecting those RNAs or those DNA segments that were bound by the protein of interest. And then, you know, sort of you keep iterating until you've basically, you know, selected the subset of DNA sequences that are, you know, that are bound. And you can do that by having a PCR amplification where those sequences that are bound are further amplified and further refined at every iteration. So using an evolutionary approach, basically. And then you can do the same with, you know, washing DNA past your, you know, factors in a very similar way. So we're going to be talking about multiple challenges in regulatory genomics, and these challenges have to do with what do I have and what would I like. So basically, there's 3 classes of objects. There's motifs, and these are the generative models that I talked about. There's targets. These are the individual instances of those motifs. And then there's the regulators, who are the ones that are binding those motifs and recognize those motifs and binding those targets. Okay? So to identify the regulators themselves, we can use homology to other transcription factors or domains.
We can use evolutionary signatures for microRNAs or experimental cloning and so on and so forth. For identifying the motifs themselves, we're going to look at de novo comparative discovery today. So the stars are the ones that are covered in today's lecture. For finding targets, we're going to look at evolutionary footprints, DNAs, TF, and chromatin immunoprecipitation. And then the dips in the chromatin profile, which we talked about last time, where at the center of a peak, there's actually a region of diminished binding, which corresponds to the specific location that's protected by the factor. So these are the three cores. And then if you have a regulator, you'd like to find the motifs that it binds. And one way to do that is you do selects or dipchip or protein binding microarrays, as I mentioned in the previous slide. And for microRNAs, you can look at their evolutionary and structural signatures or their experimental cloning. If you have a set of motifs, it's kind of difficult to find the regulator, but one of the things that you can do is basically use the sequences that you know are bound, synthesize those sequences, and use them as a bait to pull down proteins that bind them, and then carry out mass spectrometry as a way to identify the protein spectra of those transcription factors that bind these sequences. If I have a regulator, I can find its targets using ChIP-ChIP or ChIP-seq, or by perturbing these regulators and then seeing what is the gene expression change that happens as a result of that perturbation. And then conversely, if I have the targets, I can, infer the regulator through, network level analysis of enrichment. If I have the motif, I can find the targets using evolutionary signatures or using composition and folding. And if I have the targets, I can infer the motif by basically looking for enrichment in co-regulated genes or bound regions. So the slide that I showed you with ABF1, that's when you know the target exactly. But the problem that we're going to be looking at today is if you don't know the targets exactly, but you know the targets approximately, like in this 100 nucleotide region or in this 2000 nucleotide region, I know that this factor is somewhere bound. And then finding exactly where it's bound is a computationally difficult problem. All right, so that's the menu for today. So we want to recognize these motifs and understand gene regulation. And we talked about these two ways of doing it, through co-regulated gene sets or genome-wide. So let's dive into the first approach for doing it, which is building on a technique that we've seen several times before, namely expectation maximization. So the idea here is that we're going to be inferring both the motif position weight matrix and the individual occurrences of that motif, of the instances of that motif, sort of as an iterative refinement step. So here's the problem. The problem is I have a set of genes that are coordinately expressed. So all of these genes are turning on when I have galactose in my cell and turning off when I have glucose in my cell. Or they're all turning on in my brain and they're all turning off in my liver as my cells differentiate. So the question is, what drives this common regulation? And what drives this is presumably a transcription factor that actually binds each of those locations to recruit the machinery that will then turn on transcription. So the question is, computationally, given a set of co-regulated or functionally related genes, how do we find common motifs in their promoter regions? So there's many ways to do that. One way is you could use local alignment. You could basically say, okay, great, I'm going to create a local alignment, and I'm going to find short stretches of matches between these different genes. So we're going to be aligning the promoters to each other using some kind of local alignment. Well, this is great, but the motifs themselves might be very short and degenerate. So the alignment score for the local alignment might not be very strong, and most importantly, might not be very distinguishable from other locations in the genome where, you might have some spurious matches. Another approach might be to use some expert knowledge for what should the motifs actually look like. you could say, well, knowing that knowing how this factor binds DNA, it's probably a homodimer. It probably has some sort of, matching sequences that are palindromic that can be read the same way from both sides, and so on and so forth. A third way is to look for a median string by enumeration. So you could basically look for all of the possible k-mers. I just enumerate all k-mers, and then I ask, is any of these k-mers a median string that sort of matches this, commonly across all of these genes? And another way, you could basically say, let's look at evolutionary conservation, as we saw for the GAL1 promoter, the GAL4 promoter, and then let's start from those conserved blocks in the upstream regions, and then infer what are the common bound loci, basically what are the common motif patterns that are bound. So these are all sort of sequence alignment based approaches, but they all have, their own limitations, even though they're all sort of, good ways of doing this. So the technique that we're going to focus on first today is this expectation maximization approach, to basically say, well, if I knew the starting positions of my motif occurrences, it would be super simple to find the motif. I could just basically say, oh, just like for ABF1, if I have the exact start and end position for all of them, I just put them on top of each other, and I just simply count the presence of each nucleotide. Everybody with me on this one? So the second approach is, the converse of that is, if I knew the motif, I could basically simply scan each of the sequences to find the occurrences of that motif. But if I have neither of the two, maybe I can just iterate between them. So I can basically say, well, if I know the shared motif, I can infer the profile matrix. And if I have if I have the shared motif, I can infer the starting positions. And if I have the starting positions, I can infer the motif. Okay. So if I'm, given an aligned set of sequences, I can compute the motif profile matrix simply by counting at every position with what frequency do I see each of the 4 nucleotides. And maybe here we'll have the, C, A, G, G, C, A or T, C motif, okay? So that would be one way of searching through to find the, the motif. You just simply count each of those positions, and then you just add them up. But if I have the matrix, I can just simply scan and look for those systematically. So the key idea is, if I have the positions, I can find the matrix, and if I have the matrix, I can find the positions. So the key idea is, well, let's do it iteratively. We're going to define an iterative procedure for estimating both, given the uncertainty. So it's basically a learning problem with hidden variables, i.e. nuisance parameters, which I'm going to be integrating over. So these are going to be the starting positions. So let's clarify further. So we're going to have a basic iterative approach where, given some length parameter W, this is the width of the motif, and a training set of sequences, let's set some initial value for the motif and then iterate through the following. If I have the motif, I can re-estimate the starting positions from the motif. And if I have the starting positions, I can re-estimate the motif from the starting positions until convergence, until basically the change that I have in either the starting positions or in the motif is like smaller than some epsilon. And then in the end, I just return the motif and the starting positions, okay? Why does this even work? This works because when I know the motif and I scan that motif, you could say, well, if you scan your motif, you're just going to find more of the same motif. But that's not exactly true. If I have a motif that doesn't actually occur in my sequence, as I scan that motif, I'm going to find the closest match to that motif. But that closest match is going to be different than the motif. It's going to basically be, slightly altered. So that's going to be shaped, therefore, by the exact sequence of those locations. And then when I have all of these individual instances, the motif that I built from these individual instances, when I re-estimate the motif from the starting positions, that's entirely driven by the sequences that I have in hand. It's not driven at all by the starting motif that I had initially guessed. That motif gave me positions, but now I'm starting from the positions, and all of the sequence-based information is coming from that, the sequence of the sequences that I have, okay? So let's see, who's with me so far on this sort of key idea of, if I have the initial positions, I can estimate the motif, and if I have the motif, I can re-estimate the initial positions in an iterative fashion using expectation maximization or Gibbs sampling or any of the techniques that we've talked about so far. All right, so 44, 44, 13, 0, 0. All right, so let's now look at how we can actually represent the motif and the background. So we're going to assume that the motif has some fixed length, W, the weight, the width. And let's say that the width is only 3, that it's a 3 long motif, which basically matches CAG roughly. So CAG, okay? This is the most likely sequence to be generated by this. So the motif is represented by a matrix of probabilities, a matrix which at every position K gives me a character C, the probability of a character C in column K, okay? So there's that matrix. And the background is represented by simply a single column of probabilities for each nucleotide. So this is roughly a uniform background distribution. I could also have a very skewed distribution, for example, very high AT or very high GC. And I could also have dinucleotide background. And every single time, what I'm going to be asking is, how much more likely in a hypothesis comparison setup, how much more likely is it that the motif, that the particular sequence that I'm looking at was generated by the motif versus by the background sequence, okay? So who's with me so far about sort of how I'm going to be scoring using the motif, and I'm going to be scoring using the background sequence, and then using Bayes' rule to basically say, well, I have a generative model that tells me how to emit each character at every position if I am under the motif hypothesis, and how to emit each character at every position given the background hypothesis. And using these two hypotheses, I can ask which of the two is more likely to have generated the data that I'm looking at. And from that, I will then infer a parse, an annotation of my sequence. All right, so 47, 47, 700. Okay, so that's how to represent the motif and how to represent the background. We then want to know what is the starting position for that motif. So at every one of, say, 4 different sequences, Z1, Z2, Z3, Z4, I'm going to have a vector that basically tells me with what probability is the motif starting at this position here, or starting at that position here, and so on and so forth. So the element Zij of the matrix Z represents the probability that the motif starts in position J in sequence I. So this basically tells me that the motif has some, high probability of starting at that position in sequence 2, and for sequence 1 starting at that position, for sequence 3 starting at this position, and for sequence 4 starting at that position. Okay? Let's see who's with me so far on the Z vector, which represents simply the probability with which the motif is starting at each one of those locations. Awesome. So if I now have one, I can estimate the other. If I have the other, I can estimate one. So let's see how. And first of all, the responses are 60, 33, 700. So if I have the starting positions, I can estimate the motif matrix. How? Because, I can just simply scan my motif and then at every position, evaluate the start probability by multiplying across the matrix. That's how well the motif generates that matrix. And then I can do a likelihood ratio to ask how well would the background generate that matrix as well. So this is the probability of obtaining the data given that a motif. What is the probability of getting the data given that I'm in a background? So that's one part. That's sort of asking about the probability of generating that. And then once you've computed this likelihood component, that's just one part, you'd like to now ask the prior of being motif or being background. And that prior multiplied by the likelihood gives you effectively a scaling factor of the posterior, where that scaling factor is the marginal, which is the same for all of the motif occurrences. So you can just divide out the marginal. And effectively, you have a Bayesian estimation of the posterior probability of having the motif generate this particular sequence versus the background generate that sequence. So that's the ZIJ computation of computing the starting positions from the motif. For computing the motif from the starting positions, so I have now my ZIJ vector, there's three different approaches. One is expectation maximization, where all of the starting positions are weighted by their ZIJ probability distribution, and that's used to then construct a weighted average that gives me my motif. Another approach is the Gibbs sampling approach, which we're going to talk about a little bit more in detail now, which is I'm going to choose not multiple starts at every location. I'm going to choose a single start for every sequence by sampling that vector. So I have a vector that basically tells me with what probability is the motif starting at each position, and to infer the motif logo, the motif, PWM, the position weight matrix, I can just simply score each of these positions and then say, which one do I choose? And that's where these choices come together. So what expectation maximization does is that it chooses all of the starts, each weighted by the probability. What Gibbs sampling does is that it chooses single start by sampling the ZIJ vector. And the greedy approach basically chooses the best start for every sequence by maximum ZIJ. Okay? So let's talk now about this E step and then this M step. So the E step is estimating the motif positions from the motif matrix. Okay? So I have my motif matrix, I scan it everywhere, and I estimate what is the probability of starting the motif at each of those locations. So again, the posterior probability of starting at that position given my motif is simply the probability of emitting that sequence given the, that ZIJ is 1, and namely that the motif is the one that generated this, times the prior of the motif actually starting there, divided by the total evidence. So that's Bayes' rule, okay? So who's with me here on sort of this very sort of core component of estimating all these starting positions using this likelihood ratio? Lovely. Okay, so 60, 33, 700. So at every iteration, we basically calculate ZIJ based on the motif matrix. And we can calculate the probability of each starting position by simply summing things across. And to obtain that total probability, we sum over all the starting positions. And that sort of gives you this. So basically summing over all of the starting positions, you have your normalization factor. So you can assume a uniform prior, that motif, for example, is equally likely to start at any position, and then you're all set. But however, you could also use a non-uniform prior if you have some chromatin immunoprecipitation. You could use the height of the peak as your prior. Or if you have evolutionary conservation, you could outweigh the conserved locations, and so on and so forth. So you basically calculate the probability with which each position would have been generated by the motif. So at position 1, it's 0.3 times 0.2 times 0.1 times background. And then position number 2, background, motif 1, motif 2, motif 3, background. So if my sequence is GCT, GTAG. At every position, I just simply multiply through the probability of starting it at each of those. And my background distribution is 0.25 everywhere, and I can represent that here. And my foreground distribution is this CAG motif. And if I scan this across, CTG is actually a pretty good score for that. So you can see, CTG, the T gives me a low score, but the C and the T give me pretty high scores from the 0.4 and the 0.6. And then of course I normalize. Okay? So that's basically for calculating the Z vector. And then, you just scan through across and just multiply them out. And then the denominator is simply the total probability PXI, which is the same before the motif, is the same after the motif, and then is unique within the motif. And then that's, again, assuming a uniform distribution. But if you have a non-uniform background distribution, you basically can, alter these background vectors accordingly. Okay, so that's a way to estimate all of the starting position intensities from the motif matrix. Now I want to use this Zij vector to find the maximum likelihood motif estimate from those positions. So I have the starting positions and I want to estimate the motif. So as I mentioned earlier, there's three approaches for doing that. You could use greedy, you could use Gibbs sampling, and you could use expected maximization. So in this case here, where the Zij vector peaks at a single location, that basically says, well, that's probably my motif. I, seek no further. And here, all of the methods would agree. So Gibbs sampling would most likely sample this one. Expected maximization would basically sum up all the weight, and that weight is basically this. And then greedy would choose that as the maximum anyway. So all three methods agree here. In this example, where I basically have one strong peak and another smaller peak, expected maximization would basically use both in estimating the motif. And that can be problematic because it might not converge to any one of them. This might look quite different from that, even though they're the two best matches of the motif. Maybe this one matches the first half of the motif, and maybe this one matches the last half of the motif. So expected maximization might be pulled in multiple different directions and not able to converge as easily. Gibbs sampling, however, instead of saying, oh, I'm gonna, either choose always the best one or choose always the average of all of them, Gibbs sampling is basically saying, what I'm gonna do is simply choose one at random according to their probabilities. And then in this particular case, greedy, for example, would ignore most of the probability distribution. Expectation maximization would sort of try so hard to please everyone and then not converge at all. Whereas Gibbs sampling will rapidly converge to some choice because it basically says, I'm gonna, make a choice, and maybe by focusing on the end of the motif, it will be able to reconstruct the whole beginning of it. And then here, by focusing on the beginning, you might reconstruct the whole end, and you might end up with two different motifs that you might not be able to have picked up if you were using EM. So let's see, who's with me so far on the difference between expectation maximization, Gibbs sampling, and greedy in choosing these motifs? Remember, the Zij vector was computed once based on my motif, and now I'm choosing how to use the scores of this Zij vector. All right, so 53, 35, 660. And again, EM here would average over the entire sequence, which would be either very slow or no convergence. All right, so let's estimate that motif M. So recall that this matrix, which at every position gives you a character score, represents the probability of a character C in position K. The background stores the values for the background. And then at the next iteration, we have the number of occurrences of that character at that position divided by the sum over all characters, where NTK is simply the number of occurrences where, the Zij vector actually matches that motif, where the X at that position is that position of the motif. And the background is simply a uniform background distribution, which is basically tells you what is the total number of characters in your data set. So here's an example of estimating MKC from ZIJ. So basically this is the starting position estimation of the motif from, let's say in Z1, I have these starting positions. Notice that the last two characters don't have a score because this is a 3-long motif. So it has to start at least 2 before the end. So these are the scores. So a very strong score for starting here, very strong score for starting here or here, and then very strong score for starting here. And then you can put all those together and then infer what is the most likely motif. And again, it's CAG, you know, kind of like what we have expected. Okay? So what the EM algorithm does is that it converges to a local maximum in the likelihood of the data given the model. And it's a deterministic iteration with the maximum direction of ascent, and usually converges in a small number of iterations, but it's very sensitive to the initial starting point, i.e. the values of my motif. The, you know, the way to visualize this is that you have some initial set of parameters that you're starting from, and then you're hill climbing until some kind of local maximum. And then if you initialize in different positions, you know, this hill climbing will get you to, you know, sometimes a local maximum and other times in global maximum, okay? So that's for expectation maximization, where you're basically always hill climbing using all of them together. But what Gibbs sampling does is that it basically says, you know, let me just simply sample from that posterior. So Gibbs sampling is a very general procedure for sampling from that joint distribution of a set of random variables by iteratively sampling from each of them in turn. So you're sampling from one sequence, given all the others, and then sampling from the other, given all the others, and so on and so forth. And it's a very useful procedure when it's hard to explicitly express the means, the standard deviations, and the covariances across the multiple dimensions of your data set. And this is very useful for supervised, unsupervised, and also semi-supervised learning, because you specify what variables are known, and then you sample over all of the other variables. And this allows you to approximate the joint distribution based on the samples drawn, the marginal distribution based on the samples for a subset of variables, and also the expected value based on the average over the samples. And Gibbs sampling is an example of Markov chain Monte Carlo, where the sample actually approximates an unknown distribution, and the stationary distribution of that sample, you know, if you only start counting after burn-in, and if you also assume some independence of the samples, and this can be achieved by only considering in your posterior probability estimation only once every 100 samples to basically make sure that they're largely independent of each other, then that stationary distribution allows you to sort of estimate it directly from that sampling through this. And it's also a very special case of Metropolis Hastings in its basic implementation of the sampling step, but it's a much more general sampling framework. So the first application of Gibbs sampling to motif finding was by Chip Lawrence in '93, and this was a stochastic analog of expected synchronization for the motif discovery task. And it was less susceptible to those local minima because you're able to sort of only choose one and then hill climb up that one, and then choose another one. Even though it might be lower score, you might be able to sort of climb to an overall higher peak. So expectation maximization maintains that entire ZI distribution over the starting points for each sequence, but Gibbs sampling only selects a specific starting point for each sequence and keeps resampling these starting points. So given the length parameter width and the training set of sequences, you can choose a random position for A, and then pick a random sequence, estimate the probability of every starting position that the motif starts there, given the current motif positions, and then sample a new motif position, you know, until convergence, basically.
All right, so alignase is a very popular implementation of Gibbs sampling, and so is the bioprospector has also an improved version of alignase, and both use this Gibbs sampling approach of selecting random locations, computing an initial model, and then removing one sequence, recalculating the model, and then picking a new location according to the probability of that location being a motif's occurrence, and repeating until convergence. So you basically estimate this posterior probability distribution given your model and maximizing this. And you're also using pseudo counts to avoid zeros at every step of the iteration. And what Gibbs sampling allows you to do is basically not always take the maximum, but sometimes take a sideways step, which makes it less likely to get stuck at local maxima, because you might actually choose to go up this global maximum rather than just always going up to the same local maximum. All right, so what we talked about is this co-regulated gene-based motif discovery, where you can use either expectation maximization or Gibbs sampling as a way to iteratively estimate the motif from the starting positions and the starting positions from the motif using this generative framework. So who's with me so far? We basically talked about the E step of estimating the motif positions from the motif matrix, the M step of finding the maximum likelihood motif from all of the positions, and then how do we sample motif positions based on the Z vector, and how Gibbs sampling is in fact more likely to find the global maximum and actually much easier to implement than the full expectation maximization. So we have 43, 57, 00. Awesome. So let me ask about the pace. How's everybody doing in terms of the pace? Is it too fast, too slow, or just right? All right, so a few people feel that we're going a little too fast. And indeed, we are going a little too fast, but apparently you guys have totally gotten it despite being fast. So we have 11% and 50% above, and then 39% in the middle, and no one feels that we're going way too slow. All right, so that was approach number one, where we basically take co-regulated genes to discover the motifs. There's another approach which is uniquely enabled by the availability of comparative genomics. And that's basically to use evolutionary signatures for de novo motif discovery. So what is the motivation here? The motivation is that both the transcription factor centric and the region centric approaches are not comprehensive. They don't give you all of the motifs, they just give you the motifs that belong in this region with this group of genes that are co-regulated. So if I don't have the transcription factor, I will have no way of discovering the motif for that transcription factor. If I don't have the right co-regulated gene set, I might just never discover the motif that makes these genes be co-regulated. So the TF-centric approach generally requires that the transcription factor or an antibody to the factor is available, takes lots of time and money, and it also has computational challenges to discover the motifs after you have that. De novo discovery using evolutionary conservation has the advantage of being unbiased. But the downside is that it can't match the motif to a specific factor. And it also, of course, requires multiple genomes. So what is the idea for these evolutionary signatures for regulatory motif discovery? The concept is that in any one location of the genome, I might actually have a conservation of that motif instance. So if I compare multiple species of Drosophila or multiple species of mammals to each other, and I sort of look across the genome, I will see that there's stretches of conservation. And those stretches are in fact found in multiple locations where that motif matches. So if I look across the genome, I would expect to see the global conservation of that motif pattern be higher than random patterns. So we're going to basically start by looking at known motif instances and recognize that the individual motif instances are in fact preferentially conserved. And we could just take conservation islands and call them motifs, but the problem there is that
many conservation islands are due to chance or perhaps to non-motif conservation. There might be an RNA structure that's important there. So the unique signature that we're going to use is that these motifs are recurrent across the genome. And this is an approach that I used initially in my PhD thesis, and then my postdoc joined with Eric Lander, Xia Weixie, first published it in Mammals. So that was our second paper on that. And then Alex Stark, another postdoc in my lab, published this in Drosophila. Basically, we had a series of papers going through different species with increasing levels of complexity for doing this. But it all started there in yeast by sort of showing that in this GAL1 promoter, the GAL4 regulator was basically multiply bound, and these motifs were preferentially conserved. These motif instances were preferentially conserved. And then we basically said, well, perhaps we can increase our power by testing conservation across many regions. The idea was the following. We basically defined a genome-wide conservation score that allows you to basically ask, how well is this motif conserved, not in any one of its locations, but across all of its locations jointly. And then we could estimate the conservation across all intergenic regions. And what we found is that the GAL4 motif, these, CGG, 11 nucleotides from CCG, this GAL4 motif is in fact conserved 13% of the time in intergenic regions, whereas control motifs that have similar properties are only conserved 2% of the time, giving us a 6-fold enrichment and enough signal to discover the GAL4 motif as important. We also looked at intergenic conservation, basically how well is it conserved between genes, versus coding conservation, how well is it conserved within protein coding regions. And what we found, surprisingly, was that the GAL4 motif was in fact preferentially non-conserved within coding regions. So the controls were conserved 7% of the time, but the GAL4 motif was in fact conserved only 3% of the time within coding regions, indicating that there was, perhaps pressure to not bind within the protein coding regions, or simply that, the conservation patterns within protein coding regions, which has these conserve, non-conserved triplets, these bang, bang, poof periodicity, disfavors conservation of stretches of 3 nucleotides that are perfectly defined, perfectly determined. What do controls mean here? So control motifs are motifs that are generated to match the nucleotide composition, the number of genome-wide occurrences, and so on and so forth. So basically, for every kind of comparison, you want to know, well, what would I have expected? And the reason for that is that if I use only the motif sequence by itself, if it's longer, I will see less conservation. If it's shorter, I will see more conservation. But to know how much conservation I would expect, I need to construct a set of control motifs. I hope that answers your question. All right, so now let's see how many more sequences are kind of like GAL4. Basically, we know that GAL4 passes this test. It's able to be differentially discovered. What other sequences do that? So here's the first test of intergenic conservation. You basically have more, most of the motifs falling along this diagonal, where the more occurrences I have of the motif, the more conserved occurrences I have. Okay, so basically, roughly 1 every 40 occurrences is actually conserved. Okay, and I just walk along this diagonal. As opposed to GAL4, where you see these very few instances in the genome, but dramatically higher conservation than I would have expected. So that's kind of cool, but GAL4 is not unique. There's a lot of other blue points that suggest that there's many motifs that are preferentially conserved. Let's look at the second test, where basically GAL4 showed a preference for intergenic versus coding, whereas controls showed a preference for coding versus intergenic. So here, in the second test, you see that there's a lot of motifs that show higher conservation in genes more generally. But GAL4 is in fact showing higher conservation in intergenic regions than what you would have expected. So that basically suggests that there's a whole class of motifs that, instead of being more highly conserved in genes, are in fact more highly conserved in non-coding regions, which is sort of the property that we would expect for regulatory motifs. And then when we looked at upstream versus downstream conservation, so this is how often are they found in regions that are divergent from the same promoter, where both genes are, following that promoter, versus regions that are convergent, where, they're both downstream. And what we found there was that, in fact, GAL4 was indeed very highly enriched for preferential upstream conservation, but many other motifs showed preferential downstream conservation, as well as preferential upstream conservation. So that gave us a clue as to an additional class of motifs that are found downstream of genes. So remember how I was talking about earlier, there's this expectation maximization approach, there's this Gibbs sampling approach, or you could simply enumerate. So the method that we actually developed was to enumerate transcription factor candidate motifs based on their 3-gap3 conservation, the 3-gap3 pattern. And then the idea was that you could have basically 2 different sites bound on the DNA, and then we would enumerate these seeds, we would score these seeds, and then we would expand these seeds by including additional nucleotides nearby that are also conserved every time these seeds are conserved. So, and then clustering these to remove the redundancy using sequence similarity. So that basically led to a dictionary of regulatory motifs. So again, going back to this paper that I highlighted last time as well. So we basically came up with these 3 conservation tests that I mentioned earlier, where you basically have all of these preferentially conserved motifs upstream, upstream versus downstream, and then coding versus non-coding. We enumerated these seeds, these mini motifs as I call them, and then expanded those, and then clustered and arrived at these, final motif. So we expand the mini motifs into a bunch of mega motifs, and then these mega motifs are clustered into the final motifs. So this was very exciting. We basically found that we could discover a catalog of regulatory motifs completely de novo, just by looking at genome-wide conservation. And what was really exciting is that, having discovered those, we could then go back and start asking, well, does anything match this known motif, this discovered motif? And remember this ABF regulator that I mentioned earlier? So basically, CGT, GAT. So our interpretation for the top one was that this was the known ABF1 motif. This was the known REB1 motif. This was enriched in an expression cluster associated with environmental stress response. So our interpretation for that was that this is an ESR motif. Even though we didn't know it before, this basically told us that's a clear stress response motif, and so on and so forth. So some new motifs for mitochondrial genes. So basically this makes sense why they were found downstream, because yeast is using those to target its RNAs to specific locations in the cell, specifically the mitochondria. And there were some new motifs for a REB1 palindrome, a double MVP motif, and so on and so forth. So this is one way to interpret based on gene ontology enrichments, based on gene expression enrichment, and based on enrichment, in particular chromatin immunoprecipitation experiments. So again, similarity to known motifs allowed us to infer these matches both in mammals and in flies, similar to what I showed you in yeast. There were also positional biases of the motif matches. Basically, the motifs that were involved in the initiation of transcription were biased to be in the TSS and depleted in the coding sequence, and also clustered well. And then this is just a matrix of what are these functional enrichments that we saw for our motifs in different tissues of the fly. And again, we found a lot of functional clusters emerging. So this is basically telling you that we don't have to only rely on a co-regulated gene set to basically do expectation maximization and Gibbs sampling. we could simply discover these motifs de novo based on their evolutionary signatures. So who's with me so far for these evolutionary signatures for discovering motifs? So the key idea here is that instead of looking for a single occurrence of the motif, I'm going to be looking for multiple occurrences, and I'm going to be evaluating the conservation for each of those occurrences, and then clustering those motifs, basically starting from a lot of seeds, expanding them, clustering them, and then collapsing them together to basically arrive at a final set of motifs, which were now discovered de novo. And after these motifs are discovered de novo, then we're going to have to validate them using all kinds of additional functional data sets. For example, are these motifs occurring near coordinately active genes, and so on and so forth. So, 24, 59, 12, 6, 0. That's great. All right, so that's evolutionary signatures for de novo motif discovery. That's for discovering the motifs themselves. What about evolutionary signatures for discovering individual instances of those motifs? So the concept here is we now know the transcription factor binding sites. What we would like to find out is what are the individual occurrences of those motifs across the genome in order to basically be able to infer the network, the circuitry of connectivity. So how do we determine the functional binding sites of gene regulation? So the experimental approach is basically, you cross-link these cells and then you do a chip-seq or a chip-chip experiment, and then you infer the targets experimentally. But this is limited by antibody availability. It's only restricted to specific stages of development, specific tissues, and the biological functionality of most binding sites is actually unknown. And also the resolution can usually be limited. Basically, you can't identify the specific base pairs. You usually only identify general regions where those are bound. So computationally, to find targets, there's single genome approaches using motif clustering, and these require a set of specific factors that act together, but they can actually miss instances of the motif that might actually occur alone. So, in order to increase the signal, you basically have had, folks focusing specifically on combinations. But, another approach is to basically look at multiple genomes together. And these approaches usually require either perfect conservation or a very strict model of evolution. But the challenge that, you need to address is that the motif instances are not always perfectly conserved. Sometimes they're moving, sometimes they're sort of, dying and being reborn elsewhere, and so on and so forth. So, the simple case is that an instance is fully conserved in the same position, and that's great. But most of the time, or at least a lot of the time, we're finding that there's motif turnaround and movement, but there's also very far away instances and matches. So to address this, we need a new approach for finding individual motif instances. And the approach is going to be to search each of the genomes individually. To basically say, I'm going to search the human genome, the chimp genome, the rhesus genome, and so on and so forth, independently for the same CTCF motif. And then to ask, from all of the instances that I found, what is the total branch length over which that motif instance appears to be conserved? And then highlight all these branches. Notice here, the rat has a missing sequence. The cat has a missing sequence. The shrew has a missing sequence. And part of the challenge here is that these genomes were actually very lightly covered. They were not fully deeply sequenced, suggesting that many of the locations would be missing just by chance. So we didn't want to be penalizing them for those. And you can see here that the Tenrec genome has actually lost that motif occurrence. So we basically find that, for example, the CTCF motif instance at that location is conserved over this branch length, and we can just add the total branch length. And we're finding that, the total branch length score here is 2.23 substitutions per site, okay? This approach allows for mutations that are permitted by the motif degeneracy, It allows for misalignments or movement of the motifs within a particular window that can be up to hundreds of nucleotides. And it also allows you to have missing instances of motif in a dense species tree, because you can still infer the branch length from the species that are actually there. Okay? So we basically now have a way of calculating the total score at any one motif instance. So at that instance, I can simply scan for motif matches. When I find matches that pass my threshold, I can just call the motif instance as appearing in each of the species. And in the end, I just sum up the score of all of the branch lengths that have that motif there. So who's with me so far on sort of this individual scanning of all of the species, then adding up their branch length? All right, so we basically have 19, 44, 31, 6, and 0. This is great. Most folks are following quite well. So what's the next challenge? The next challenge is now that for a very long motif, I should be very excited that I have a very long branch length. But for a very short motif, I probably shouldn't be too excited, because short motifs can just be conserved by chance. So what I want to do is convert my branch length score into a confidence score. Basically evaluate the chance likelihood of a given score, whether that sequence could also be conserved due to, say, overlap with an unannotated element, such as a non-coding RNA. I want to be able to account for differences in motif composition and length. For example, a short motif might be likely to be conserved by chance, but at the same time, a very AT-rich motif might be conserved by chance in an AT-rich genome. So what we'd like to do is construct a set of control motifs, just like before, where we can take our original motif and then produce hundreds of shuffles of our original motif, just shuffling the positions, and then filtering these motif shuffles and requiring that they match the genome within 20% of the original motif. Not that they're conserved within 20%, but that they match the genome within the same rough frequency. And the reason for that is we want to sort of avoid places where the shuffle had a very different dinucleotide composition that was excluding it from the genome, and so on and so forth. So we then sort the potential control motifs based on their similarity to other known motifs. We cluster these potential control motifs and take at most one from each of the clusters in increasing order of similarity to the known motifs. So basically, we're going to choose highly dissimilar from the known motifs, and then one per cluster to avoid biasing ourselves to always test the same motif over and over again. So we now have these, branch length score, and we now have a lot of control motifs, and we'd like to know how well are these control motifs matching the branch length score distribution that I obtain for the real motifs, okay? And that's what gives us this curve here. At increasing branch length scores, I have decreasing numbers of motif occurrences that achieve that score. And we can use the motif-specific shuffled controls to determine the expected number of instances at every branch length score due to chance alone or due to non-motif conservation. So this gives us this light blue curve here, which is the noise. Basically, our actual motif, CTCF, occurs at this level, but the shuffled CTCF occurs at lower levels of branch length score. So what we can do now is compute a confidence score as a fraction of the instances over the noise at a given branch length score, giving us 1 minus a false discovery rate. So that's the confident, which basically says that if I'm all the way here, 90% of my instances are from the signal and only 10% I would have expected from the noise. But if I'm all the way down here,
I would have expected, I observe 3,500 instances. I would have expected 2,000 instances. That's basically suggesting that about 40% of them are actually real. So my confidence is only 40%. So who's with me here on sort of how we can use confidence
to basically obtain a much more interpretable signal for each of those motif instances? So instead of having a branch length score, which will depend on the length of the motif, the nucleotide composition, the occurrences, and so on and so forth, we now have a single confidence that takes all of that into account and then corrects for that. We have 38, 31, 31, 0, 0. That's great. So that's the confidence score. And the advantage of that is that it works for all motifs. It doesn't matter if it's a long motif, it's a short motif. We can just use this metric of confidence as a common metric that is applicable to all motifs regardless of their composition. So we can now basically ask, what is the enrichment in, say, background versus foreground, and so on and so forth. So basically what we can do is ask, if I'm increasing my confidence for transcription factor motifs that I'm discovering, then where are these instances happening? So basically, remember, this is about finding individual occurrences of this motif across the genome, and we can use the confidence tuning to deal with short motifs or long motifs in the same way. So at 80% confidence, to get 80% confidence with a long motif, I just need a few species. To get at 80% confidence with a short motif, I need many, many more species. Okay, so that's where confidence comes in. But now at a given confidence threshold, I can basically say, where are the instances that are 80% confidence occurring in the genome? And what you can see is that as I'm increasing the threshold of confidence that I'm requiring, these motifs are increasingly occurring in promoter regions and 5' UTRs, but to a much lower extent within protein coding regions or introns, which are sort of, disappearing very rapidly, and 3' UTR that just completely disappears. So transcription factors appear to be preferentially occurring in the 5' UTR and in promoter regions. By contrast, if you take the same exact score and you apply it to motifs that are matching microRNAs, then you see that with increasing confidence, I'm increasingly selecting for 3' untranslated regions where microRNAs are known to be bound, and decreasing and selecting against promoter, 5' UTR, CDS, and so on and so forth. Okay? So you can now discover a lot of motif instances, and now you'd like to validate those. So you can use independent experimental evidence or look for functional biases and enrichments. So one very cool way of doing that is to basically ask, what is the strand bias? Is the motif occurring this way or that way with respect to the gene start? So what we find is that transcription factor motifs are, don't have a preference. As you increase the significance threshold, the confidence threshold, you basically see that they're roughly half occurring the forward strand versus half occurring the reverse strand. Whereas microRNA motifs are non-flippable. And the reason for that is that they are recognized at the RNA level, where the microRNA will bind your target mRNA and hybridize to it. You can also see this increased specificity and sensitivity using the BLS. You basically see that, if you require full conservation, you capture many fewer motif instances, but with branch length score, you capture dramatically more motif instances. And you can also see that these motifs are in fact matching very well with known bound sites using experiments. So basically at 50% confidence, you basically dramatically increase your signal for your enrichment in chromatin immunoprecipitation experiments. So that suggests that the individual motif occurrences are able to predict where the transcription factor will bind prior to actually doing the experiment. Okay? And then with increasing confidence, you basically have increasing conservation of those motifs. So if you basically have both human and mouse, then that is dramatically enriched. And you can see that for individual factors and individual motifs. And what that allows us to do is now start building regulatory networks that allow us to connect transcriptional regulators to their individual targets across the genome, and then infer these networks, infer individual edges, and then validate those through the literature, but also experimentally. So in the next lecture, we're going to talk about how we can combine these networks to now start analyzing properties of the inferred networks. So we're not going to cover that grayed out section, but what we talked about today is what are regulatory motifs? How do we discover these motifs in co-regulated gene sets using expectation maximization and using Gibbs sampling? And then how do we discover motifs de novo across the entire genome using this genome-wide conservation signature, and then motif enumeration and motif extension? And how do we validate the discovered motifs? Again, this is the generative model that has many instances across the genome. And how do we use evolutionary signatures for discovering individual motif instances and using the phylogeny of the species that are connected to infer the branch length score, and then translating this branch length score into a confidence score using these control motifs, and what branch length score distribution do we expect for those control motifs. All right, so who is with me so far on the whole lecture? Who feels that they are following all of these different parts? This is great. So we have 7%, 64%, so on and so forth. So this is changing slightly. So 12, 53, 24, 6, 6. And then last question is, who feels that they've learned stuff today? Awesome. So we have 65% of people in 4 and 35% in 5. All right, so thank you, everyone. And remember, project proposals tomorrow, and link the Google Docs directly from the weekly feedback page. And then make sure that you enter your names so that the folks who don't have groups yet can easily find them. And so if you can have them by the mentoring session, that would be best. So if you can have them by 4 p.m., that would be great. It doesn't need to be final, but make sure that it's there and that we can go through it and click on it and start looking at them, because we're going to be using that to guide the peer review of those proposals. Okay? All right. Thanks, everyone. See you tomorrow. Bye-bye.